Hi, I'm Yesh Doctor. I'm a graduate student in Prashant Mali's lab here at UC San Diego, and I'm really excited to talk to you guys today about the work we've been doing in PerturbSeq, and specifically trying to do genotype-phenotype mapping at scale. So broadly speaking, what we're interested in is genome interpretation through genomic perturbations. So the way we do our experiments is really in three axes. One is we try to knock down or perturb a set of genes using CRISPR-Cas9. And specifically in our application, we're using CRISPR-Cas9 fused to an inhibitory domain called CRAB. And so what we've done is we've targeted 200 chromatin modifying and metabolic enzymes and try to knock them down to fully understand their function in terms of regulating the cell state. And so we do this across two different cell types. I'm going to talk today about the COLF 2.1J stem cell line, but we've also repeated this experiment in the MDA-MB468 breast cancer cell line. And so across these two axes, what we do is we take our cells and we run them through single cell RNA sequencing to try to understand what effect does the genomic perturbation have on the cell state or the phenotype of the cell. And so through single cell RNA sequencing, we're able to generate these matrices for every perturbation we make or every gene knockdown we make. So per perturbation, we get a cell by gene matrix, where for each cell that was perturbed by that guide, we get the set of genes and the entire transcriptome of that cell. And so by looking at these matrices, we hope to infer the role that each gene has in regulating the cell state. And so fundamentally, how we actually do this in an experimental sense is through a pooled single cell screening approach. And so this is known as CRISPR or CRISPR PerturbSeq. And so generally in this approach, I'm going to show the schematic for the MDA first and then move to the COLFs, is that we take our cells and we transfect them at pretty low MOI with a virus that contains our guide RNAs that target each gene. And so in this experiment, for 108 targeted chromatin regulating genes, we had 6 guide RNAs per gene, as well as including non-target control guides as well as a reference for an unperturbed cell. And so once we transfect our cells, or transduce our cells rather, we then, in the MDA experiment, also treat the cells with different drugs, including DMSO, paclitaxel, and vorinostat, to try to understand not only what do the perturbations do, but what do these drug perturbations do on top of this. And so once this experiment is run, we'll place these cells into our single cell pipeline and then try to analyze them downstream. But today I'm going to specifically focus in on the COLF dataset, which is a little bit simpler to understand. And so in this case, all we do is we take our COLFs that we've engineered to actually express this dCas9, and then we transfect them or transduce them with our virus that targets these 108 chromatin regulators, and then we go on to single cell RNA sequencing. And so like I mentioned, that's going to be my focus for the talk today. And so just zooming in, taking a closer look at exactly the experimental design we used, once we transduce these cells with a virus, they're passed into our single cell approach, and then from that, we generate cDNA as the output. And from this, each guide RNA is actually barcoded as well. So we can get for every cell the associated perturbation and the entire transcriptome, or the gene expression library and CRISPR library. And from this, we use next generation sequencing to get a readout. And so for those of you who are not familiar with what goes on in the single cell process, here's kind of a zoomed in view of what's actually going on. And so, fundamentally what you need to know is that this is a technology pioneered by many companies, but one of the key companies is 10x Genomics. And they invented a sort of droplet-seq or drop-seq method where through a microfluidic channel, each cell gets passed into a droplet of oil. And inside of this droplet, there's a single cell and a bead containing a bunch of oligos that help us to identify what cell was in that bead. And so what cell was in that droplet. And so all of the RNA that's inside of that cell actually gets labeled with the exact same barcode, genetic barcode. And so we can, from that barcode, infer that this was a single cell, and that's how the single cell approach works. And so this is a very dense slide here, just showing the entire work through. What I just went through with you guys is the experimental workflow that we use, but there's a whole other computational workflow that's required to actually be able to even process this data. And so I don't intend to go through every step of this, but I think this is a nice reference when you're looking later on the open house to see exactly what the data you're getting is. And this data has actually gone through several steps of cleanup and processing before it's being provided to you guys. And I'm going to give a high-level overview in the next slide of the type of computational cleanup that we do. But just want to mention that the tools we use are all highlighted in this web portal called Single Cell Best Practices that uses tools from ScanPy, Seurat, and PertPy. So you can actually go and look this up as a reference to see the techniques we use, and we've adopted them from this group. But just, at a high level, what you're looking at is once we get the data back from sequencing, we first have to aggregate these channels, right? So what does that mean? So when we do our single cell approach, in the 10x genomics, there's a chip where you load your cells into, but we cannot fit all the cells we require into a single channel of this chip. So actually we have to aggregate together a bunch of these channels. And so there's a tool called Scanorama, which really is a computational tool that assists in doing that. So once these channels are aggregated, we then do what's called dead cell QC or dead cell quality control. Basically here you want to remove any cells that potentially could be dead. And so one good proxy for this is having a high percentage of mitochondrial reads indicates that the cell might actually be dead and not be a viable cell to study. So we basically filter for only cells that have a low percentage of mitochondrial reads, indicating that they're likely live cells. And then you go into ambient RNA correction, which basically corrects for any floating RNA that might be contaminating your sample, and doublet detection. And doublet detection involves the fact that, this technology isn't perfect. Sometimes within one oil drop, you can get two or more than two cells in a single oil drop, but they all get the same barcode, so they're read in as a single cell. And so obviously this can cause many data errors going forward. So there are computational tools that help to identify doublets and try to scrub these out of the data set. Then we go into guide RNA assignment. So the key idea here is that for every cell, we have the transcriptome, sure, but we also have the CRISPR library. And we want to use the CRISPR library to infer what perturbation did that cell get. And so that's a single guide RNA assignment part of our pipeline. And then once we've assigned cells and only taking cells that got a single guide, not zero guides, not more than one guide, we then go on to feature selection. So the point here is that, we're detecting on average over 5,000 genes per cell, and if all the genes we collect in our reference, there are 36,000 genes, including non-coding genes. But, not all these are very informative, right? A lot of these are exactly zero counts across the entire data set. And so really what you want to do is you want to subset down to a set of genes roughly in the 6,000 gene range that are actually informative for your data for downstream clustering. So once you do feature selection, we also have to do confounding effect removal. A lot of the confounding effects in single cell can simply be traced back to what phase of the cell cycle each cell is in. And so actually by regressing out cell cycle or regressing out the total number of reads or counts per cell, you can actually start to see signal that's only associated with your perturbations that start to emerge. And then, so finally, what we do is we actually use Mixscape, which is a tool developed by the folks who developed Seurat, to identify only cells that actually got perturbed, right? So sometimes we can sure get a guide into a cell, but it may not have actually knocked down that gene to a significant level. So here we're trying to filter for only cells that actually were perturbed, or the gene we were targeting with our guide actually got knocked down. So once we've gone through all this quality control, we can finally start to see some signal emerge. So the first thing I want to remind you of is that the data you're looking at here is in the iPSCs and looking only at knocking down 100 chromatin regulators, right? So first, what you want to look at is a sanity check. You want to say, did our screen actually work? And so one quick way to check for that is by comparing cells that received non-targeting control guides, or these are guides that don't target anywhere in the genome, and comparing the expression of the genes that were also targeted in the screen. You should expect to see a knockdown when we actually use a targeting guide RNA. And sure enough, across our data set, we see a significant knockdown of the gene targets we were trying to perturb, right? So that tells us, okay, likely our screen did work. And then what we do is we start to look at the number of differentially expressed genes per perturbation. And we're able to actually see that by knocking down genes, we start to see differentially expressed genes across cells. And we start to see cell states perhaps moving, right? So one example of cell states perhaps moving to different places are with these two genes, DNMT1 and UHRF1. By knocking down these genes, we see that cells tend to cluster in this bottom left region, and not many NTC cells actually cluster in this region, telling us that we perhaps at a gross level change the cell state, right? And then what we also are able to do is create a perturbation correlation network. And this is actually exactly what you're going to do in the open house session is take the data and go through the code and actually make this plot that you're looking at right here. And so essentially what this plot is showing is that for each perturbation, what we've done is computed the mean of all cells, because for each perturbation, you can have 50 cells. So you want to just shrink it down to a single vector. So we call this the perturbation mean. And then we want to compute the pairwise Pearl-Lacing correlation matrix between all perturbation means. So fundamentally what this means is how closely related are the phenotypes of cells receiving two different perturbations, right? So if two perturbations induce a similar phenotype, they'll have a high Pearson correlation, right? And so then, but this, you can imagine this matrix is pretty large and it's still 100 by 100, so it's hard to exactly visualize what perturbations cause similar phenotypes. So then we actually run UMAP on the correlation matrix to help us visualize which perturbations correlate similarly. And so here is where we create this plot. And what you can start to see is actually there are clusters in this plot that actually have known biological meaning. So just to go into some examples, one is that we notice that histone acetyltransferases on the left here and histone deacetylases on the right tend to cluster closer to each other, right? And then looking more at what we know in literature, I'm just going to go to the DNMT1 and UHRF1 example. Actually, in literature, we know that UHRF1 recruits DNMT1 to DNA to create chromatin modifications. And so it was very validating for us to see in this screen that actually these two correlate quite well in the PerturbSeq space as well, showing that perturbing these two genes also induces similar phenotypes, which is expected based on the literature, right? And so through the open house, I encourage you guys to look at some of these other clusters that you might start to see. And yeah, I hope you guys find some very interesting biology. Thanks.